Hello everyone and welcome back to Neuropsychology. So in the last video we talked about everything in the visual system, or the main characteristics at least. We talked about the anatomy of the eye, the sensory receptors in the retina, which we call the photoreceptors, and these are rods and cones. We talked about the optic nerve and the optic chiasm and the optic tract, and then we talked about the two major visual pathways into the brain. So in this video, we are going to talk about audition, so how we hear. We're going to talk about pressure waves, we're going to talk about the anatomy of the ear, and how the ear transduces sound into neural signals, and we're going to talk about tonotopic representation and auditory pathways in the brain. Okay, so audition is the ability to construct perceptual representations from pressure waves into the air. So the auditory system is very complex, and technically for two reasons. So first, many transformation of pressure waves actually take place within the ear before action potentials are generated in the auditory nerve. And secondly, the auditory nerve projects to many different targets in the brainstem and the cortex. So even though it projects to many areas, we are only going to cover the major features in this lecture. So the human auditory receptors respond to sound wave frequencies between 20 and 20,000 hertz. And other animals can hear different bands of frequencies. So for example, elephants, they can hear below 20 hertz. And a bat can create sounds as high as 120,000 hertz. And also when people get older, they will lose high frequency hearing. Okay, so remember the slides from 2 videos back? So I technically already went over this slide, but I want to talk about it again. So remember that sensory receptors transduce energy into action potentials. Well, in addition, air pressure waves are first converted into mechanical energy, and then that activates the auditory receptor cells and in turn will produce action potentials. So here you have a very schematic picture of the ear, how pressure waves go into there. They make the eardrum vibrate, then the little ossicles, and then eventually it goes into the cochlea, and then it goes to the brain. So let's dive a little deeper into this. So changes in air pressure cause sound waves. So here you see an example of that. Here you see more pressure, less pressure, more pressure, less pressure. You can technically draw that up just like a visual wave, like this. So here it's high pressure, low pressure, high pressure, etc. So the frequency, amplitude, and complexity of these changes determine what we hear. So we hear the frequency or speed of pressure changes as pitch. So it can be high pitch or it can be low pitch. High frequency would be when these high pressure points are close together and occur more often. So we hear the amplitude or intensity of pressure as loudness. And then lastly, we hear the complexity of pressure changes as timbre. So technically it's the perceived uniqueness of tonal quality of a sound. So that's why notes C on a piano and a C on a guitar, they're the same notes, but they sound different. That's because of the timbre or the complexity. So receptor cells in the inner ear detect these differences in air pressure and convey them to the brain as action potentials. Areas of the cortex in the temporal lobe interpret the action potentials either as random noise or they'll interpret it as sound, like for example music or language. Okay, so let's go over this a little bit more. So here you can see how we hear the sound differences in pitch, loudness, and timbre. So low frequencies would be a low pitch sound and high frequencies would be a high pitch sound, as you can see right here. And when there is a high amplitude, so when these are larger up and down, when there's a high amplitude in a sound, it sounds as a loud sound. But if there's a low amplitude in sound, it's a soft sound. And then we have the timbre, which is the complexity. So here you see a simple sound wave. Here you see a very complex sound wave. And timbre gives us information about the nature of the sound. So for example, when you hear someone play a guitar or on a piano, like I just said, it's the same note, but it sounds different because of this complexity. Okay, so let's go over what all the parts of the ear. So there are three main anatomical divisions of the human ear. We have the outer ear, which is all the way from the tympanic membrane or the eardrum to the outside of the ear, which we call the pinna. Then we have the middle ear, which are the ossicles. And then we have the inner ear, which the main player is the cochlea. So let's go in a little bit more detail of this. Okay, so here again, you have the outer ear right here, middle ear right here, and inner ear. So the outer ear consists of the pinna, which is your external structure or your ear. And it catches wave of air pressures, and then it directs it into the external ear canal, which you can see right here. So this is the external ear canal or the auditory canal. And then this auditory canal amplifies the sound somewhat and then redirects it to the eardrum. So the tympanic membrane is also the eardrum. Same thing, different names. So when these sound waves strike the eardrum will start vibrating at frequencies varying with the wave's frequencies. So on the inside of the eardrum is where the middle ear starts. So the eardrum is technically still part of the outer ear. So in the middle ear, it is technically an air-filled chamber, and it contains the three smallest bones in the human body. So these are what we call ossicles. Aren't they cute? Three smallest bones in our bodies. So within the ossicles, we have three parts. We have the hammer or malleus, we have the anvil or also called incus, and we have the stirrup, which is also called stapes. So don't get confused. I know there are two names for everything in neuroscience. And it gets very confusing when all our body parts have so many different names, right? Okay, so what I want you to remember is that the malleus is connected to the eardrum. And then the stapes is connected to what we call the oval window. So technically, the smallest bones in the human body connect the eardrum to the oval window. Okay, and then we have the inner ear, and the inner ear consists of the cochlea, which contains the auditory sensory receptors, which we call hair cells. And then the cochlea is kind of rolled up like a snail, as you can see right here. And we're going to dive deeper in that in the next couple of slides. Okay, so here you see a similar picture as what we just saw, and similar information as what we just went over. So let's go over it. The pinna catches sound waves and deflects them into the external ear canal. So the pinna takes all these sound waves and deflects it into the auditory canal right here. Waves are amplified and directed to the eardrum, causing it to vibrate. And then, in turn, that vibrates the ossicles. Then the ossicles will amplify and convey vibrations to the oval window. So the cochlea is connected to the oval window, and the cochlea is rolled up into the shape of a snail shell. So it is filled with fluid, and in the middle of the cochlea, there is this membrane called the basilar membrane, which is technically just floating in there. So the hair cells are embedded in parts of the basilar membrane, which we call the organ of Corti. Okay, so let's take a look. I know I went over a bunch of terms, so let's look at all of those. Okay, so we went over the first four, so let's go over number 5. So vibrations in the oval window sends waves through the cochlear fluid, which is the fluid inside of the snail-like structure. Then it causes the basilar and tectorial membranes to bend. So here you have the basilar membrane, here you have the tectorial membrane. And then in the basilar membrane, you have all these little hair cells, right? So here you have inner hair cells, and here you have outer hair cells. So when the basilar membrane bends, it causes the cilia of the outer hair cells, which are these tiny little mini hairs on the hair cells, to bend. And this bending eventually will generate an action potential or neuroactivity in the hair cell. So I know that was a lot of information, so feel free to pause the video and look at these images one by one again. Okay, so the frequency of a sound is transduced by the longitudinal structure of the basilar membrane. So if you unroll this snail-like structure, the cochlea, the basilar membrane is simply just a sheet of tissue. The basilar membrane is narrow and thick at its base. So this is near a round window. And then it gets thinner and wider at the apex within the cochlea. So as you go inwards, it gets thinner and wider. So higher sound frequencies cause maximum peaks near the cochlear base, and lower sound frequencies cause maximum peaks near the apex. Okay, so different points on the basilar membrane represent different sound frequencies. So this is technically what I mean with tonotopic representation. So this also applies to the auditory cortex, so the cortex in the brain, where different locations also represent different sound frequencies. So this is very similar as in the visual system. So in the visual system, each receptor cell has a certain receptive field. In the auditory system, each auditory receptor cell has a receptive field as well, and so does each cell in the higher auditory centers. So the receptive field of a hair cell is not a point in space, but it's rather a specific sound or a band of frequencies. Okay, so let's talk about the auditory pathways into the brain. So the axons of the hair cells, they will leave the cochlea to form a major part of the auditory nerve, which we call the 8th cranial nerve. So this nerve first projects to the level of the medulla in the hindbrain, synapsing either in the dorsal or ventral cochlear nuclei or in the superior olivary nucleus. The axons of neurons in these areas will form the lateral lemniscus, which will terminate in the inferior colliculus in the midbrain. Remember one of the first videos when we were talking about the midbrain, where the inferior colliculus was? If you forgot, please go back to those videos. So the inferior colliculus is also connected to the superior colliculus, and they work together. The inferior colliculus is very important for sound localization, and the superior colliculus is very important for orienting the head towards the sound direction. So again, they work together. Two distinct pathways emerge from the colliculus and course to the medial geniculate nucleus in the thalamus. So remember in the previous video when we were talking about vision, the visual system also goes through the thalamus, but in vision it goes through the lateral geniculate nucleus, so the LGN in the thalamus. And in addition, it goes to the medial geniculate nucleus, or the MGN in the thalamus. So don't get those two confused. So after the projections to the MGN in the thalamus, the ventral region will project to the core auditory cortex, or the primary auditory cortex, or A1. And again, here A stands for auditory and 1 for primary. And the dorsal part of that projection will go to the secondary auditory regions. So there are multiple independent ascending pathways into the cortex, and here we technically just cover 2 of them. Okay, so I know I just gave you guys a lot of terms, so here's a little recap of everything we just talked about. So, sound waves are pressure waves, and first they go into the ear through the pinna. The pinna will then send it through the auditory ear canal right here, and eventually it will hit the eardrum. and then the eardrum starts vibrating. When the eardrum starts vibrating, then the ossicles, which are the smallest bones in our body, will start vibrating as well. And then eventually it will go into the cochlea. When these vibrations hit the cochlea, these hair cells will register that. They will bend. These little cilia or mini hair cells on the hair cells will bend. So this eventually will lead to an action potential, and the axons of the hair cells will form the auditory nerve. Then the next stop is the superior olivary nucleus, which is right here. Then we have the inferior colliculus. Then we have the medial geniculate nucleus, and then it goes to the auditory cortex. Okay, so try to remember this picture. This picture makes it a little bit easier. It goes through the ear, then it goes through all these structures, through the cochlea. Here you see the right side in blue and the left side in red. So in the cochlea, you have these hair cells, and when they bend, they will form action, or they will create action potentials. The axons of these hair cells will go to form the auditory nerve, which will go to the superior olivary nucleus. Then it goes to the inferior colliculus, then to MGN in the thalamus and auditory cortex. Okay, one thing people always get confused with is that the first step from the ear to the brain is superior olivary nucleus and then inferior colliculus. These terms are always confusing to people, so definitely memorize those well. And that was the end of this video. I will see you guys back soon for the next video.